I just wanted to start by thanking you so much for the invitation to speak tonight, today, wherever you are in the world. It may be a different time, but thank you so much for the invitation to speak and for all the wonderful things you've done for starting this Beck-Fahrner Syndrome Foundation, and for all that you just have done for the foundation and for really all individuals with this condition. It's really quite amazing in such a short period of time. And I'm so happy to be here with everyone who has joined virtually. I'm happy to meet everyone and excited to share the evening with you all. So tonight, I'm going to just give basically an overview of Beck-Fahrner syndrome, which we previously called TET3 deficiency. And so I'll just get started. I have no disclosures. So the objectives for tonight are to introduce the concepts of genetics and epigenetics, to understand Beck-Fahrner syndrome as a genetic disease, to understand epigenetic consequences of Beck-Fahrner syndrome, or in other words, deficiency of TET3, to share knowledge of molecular changes in Beck-Fahrner syndrome, to inform you about the clinical features of Beck-Fahrner syndrome, and to introduce ongoing research in our novel and multidisciplinary epigenetics and chromatin clinic at Johns Hopkins, where we specialize in providing care to individuals with Beck-Fahrner syndrome and related disorders. So I just want to start by introducing some basic concepts in genetics. So our bodies are made up of billions of cells, and each cell contains genetic material in the form of DNA. Our DNA contains about 22,000 genes, and each gene determines specific traits. So things like eye color, how we grow, how our brain develops. Each gene is a set of instructions or a code to make a protein, and each protein has a specific function. The DNA consists of 4 bases. It's really this DNA sequence or code, and the bases are shown here. So if you look over here to the right, here is a schematic of our DNA. You can see that it's divided into these functional units called genes. And if we look at this gene here in this pink color, we're sort of zooming into that gene and seeing that the gene is really made up of this DNA sequence or code, which consists of these 4 bases, A, T, G, and C. And each gene, its job is to make a protein that has a specific function in the body. And the term genetics really refers to this DNA code. So TET3 is just one of the many genes that we have in our whole collection of genes, which we call the genome. And the TET3 gene makes a protein, which is also called TET3. And TET3, the protein, is an enzyme that removes or erases a particular chemical mark on the DNA called DNA methylation. And I'm going to get to that in a little bit more detail later. So genetic changes in the DNA code cause disease. And when you have a genetic change in the TET3 gene, this causes Beck-Fahrner syndrome. So changes in the DNA code that cause disease are called variants or mutations. And these are spelling errors in this DNA code, and they can be missing bases, extra bases, or incorrect bases that get incorporated into the DNA. And when this happens, you get a protein that has an altered function. It can have a reduced function. It cannot function at all. There can be no protein produced. It kind of depends on the different type of mutation. But basically, in the case of TET3, you have a variant or a mutation in the DNA code, and that leads to the TET3 eraser not functioning properly. And it may have a reduced function, or it may not function at all, or maybe no protein is produced, again, depending on the type of mutation. And that's what causes Beck-Fahrner syndrome. So I think many of you are probably aware that individuals have 2 copies of each gene, including TET3. And in most cases of Beck-Fahrner syndrome, the condition is autosomal dominant and results from variants in a single copy of the TET3 gene. So even though there are 2 copies of the TET3 gene, most individuals with Beck-Fahrner syndrome have a mutation in just 1 copy, and that leads to the TET3 eraser not functioning properly. But remember, there's this other copy that doesn't have a variant or mutation and is functioning properly. And that is how most cases of Beck-Fahrner syndrome present. So these individuals then, their variants are usually not inherited from an affected parent, but usually this type of mutation occurs first in the affected child or the affected individual. Occasionally, though, a parent might have had similar features or more subtle features and can actually have the same variant in TET3, and that they have then passed down to their child through no fault of their own. But for the most part, individuals with Beck-Fahrner syndrome have a new change in 1 copy of their TET3 gene that causes the disorder. Rarely, though, there have been cases reported of Beck-Fahrner syndrome that may be autosomal recessive, or that we have thought were autosomal recessive. And in these individuals, they've been found to have variants in both copies of the TET3 gene. And so in this case, you would have a variant in TET3 on both copies, and that means both copies of the TET3 eraser protein would not be functioning properly. And this is another way that individuals can get Beck-Fahrner syndrome. Now, in most cases of this type of Beck-Fahrner syndrome, we often will see that a parent might have more mild symptoms, or at least one parent might manifest more mild symptoms. So what does the TET3 eraser do then, and how does this cause disease? So we do not know for certain how mutations or variants in TET3 cause disease, but we do know that TET3 erases this special mark on the DNA called DNA methylation, and that this mark is important for properly switching off genes. And so I'm going to talk a little bit more about this, but we have shown that individuals with Beck-Fahrner syndrome have an increase in this mark. And so one thought is that this altered DNA methylation mark, or the inability to erase it at many genes, may lead to disease. So let me show you what I mean. So again, as we talked about before, DNA is our genetic material, and it is divided into functional units called genes. Here's gene 1 and gene 2. And each of the genes determines specific traits. The DNA doesn't just exist as a single sort of strand in the cells, but instead it is wound around these proteins called histones. And it's the histone and the DNA together that is called chromatin. Both DNA and histones can be marked with chemical groups called epigenetic marks. And these epigenetic marks can really determine which genes are turned on or off in particular cells. And the epigenetic mark of interest here is called DNA methylation. And DNA methylation and other epigenetic marks, as I said, switch genes both on and off. And the presence of DNA methylation here at the beginning of a gene turns genes off. And I've indicated that here with the word off, and by having the gene have a red color, which means stop. It's not turned on, it's not expressed. The absence of the DNA methylation actually allows for a gene to be expressed, and so the gene is turned on. So gene 1 is colored in green to show that it's on or expressed. So these particular epigenetic marks, and here DNA methylation, are placed and removed by enzymes that are collectively referred to as the epigenetic machinery. Epigenetic machinery consists of enzymes that place marks that we'll refer to as writers, and enzymes that remove marks that we'll be referring to as erasers. So writers and erasers of particular epigenetic marks, here DNA methylation, are present at particular genes and are important for properly switching these genes on and off. So again, DNA methylation switches the gene off. Lack of DNA methylation switches it on. Specifically, the TET enzymes, of which TET3 is a member of this group, remove DNA methylation, and a different set of enzymes place it, but we don't need to worry about what those are called at this point. And because TET3 is an eraser of DNA methylation, we thought that when TET3 is reduced or not functioning properly in Beck-Fahrner syndrome, indicated here by having a broken eraser, it cannot fully remove DNA methylation at target genes like gene 1. And so we tested this and found that DNA methylation in the blood of individuals with Beck-Fahrner syndrome actually is increased. So suggesting that this theory that TET3 cannot remove DNA methylation may in fact be true. And the increased DNA methylation may abnormally turn genes off that are supposed to be on. And this could not only affect those direct genes that the DNA methylation is normally present at, but it could also indirectly affect other genes, causing changes in many genes and whether or not they are turned off or on properly. And we think that it's these changes in gene expression that may be the cause of Beck-Fahrner syndrome. So just to visualize a little bit more what I mean is when you have a functioning TET3 eraser, TET3 removes DNA methylation and switches on genes 1, 2, and 3, shown here in green, but it may not affect genes 4 through 8. In the setting of Beck-Fahrner syndrome, however, when TET3 is not functioning properly or when there's a deficiency of TET3, genes 1, 2, and 3 may be incorrectly switched off. And if gene 1 is involved in proper learning and neurological development, gene 2 is involved in proper growth, and gene 3 is involved in the development of the head and facial structures, these are the processes that will be disrupted and the features that will result. So what are the clinical features of Beck-Fahrner syndrome? So we know that TET3 had previously been shown before we knew it caused this disorder. It had already been shown to be important for proper brain functioning. And so it is not surprising that many features of Beck-Fahrner syndrome are neurological in nature. And these include developmental delay, and this can affect gross motor skills, fine motor skills, and speech and language, intellectual disability, specific learning disabilities, hypotonia or low muscle tone, autistic features and or social difficulties, ADHD, anxiety, depression, seizures, and other movement disorders and atypical movements. I'm not going to say anything more about these neurological features because Dr. Harris will probably be expanding on these features. But I do want to tell you about some additional symptoms that have been noted that are not neurological, but that affect other systems in the body. And these include growth differences. So individuals with Beck-Fahrner syndrome can have overgrowth or poor growth, and this can affect either the height and or the head circumference. So they can have a larger than expected head size, or can be taller in stature than expected for age, or the reverse can be true. They can be shorter than expected in stature, or have a smaller head size than expected. Other manifestations include eye findings, and these include vision impairment, strabismus, which has to do with alignment of the eyes, and nystagmus, which has to do with abnormal movements of the eyes. They can also have hearing loss, and this is a newer feature that we have more recently kind of noticed, and this can be conductive in nature or can be related to recurrent ear infections. They can have musculoskeletal findings, including joint hypermobility or joint laxity. And one example of this is hip dysplasia, which can occur in the newborn period. They can have spine curvature, which includes both scoliosis, which is sort of an S-shaped curve of the spine, or kyphosis, which is sort of when the back is sort of hunched over. Individuals can also have gastrointestinal problems, including feeding difficulties or constipation. They can have cardiovascular findings, including valve differences. So as many of you probably know, the heart chambers are connected via valves, and there can be differences in the way the valves form and function. They can have differences in the connections between the various chambers of the heart or septal defects, or they can have more complex abnormalities. And then rarely individuals can have genitourinary findings, including cryptorchidism or hypospadias, which refer to undescended testicles in the case of cryptorchidism, and hypospadias refers to differences in the way that the penis is formed in males. And they can have enlarged kidneys as well. Oh, and I'll just point out that it is really important to know that not everyone with Beck-Fahrner syndrome will have all of these symptoms. With any genetic condition, affected individuals will have some, but not all features. And it is important to know that we may not know all of the features of Beck-Fahrner syndrome yet, because this is a new disorder that we just described in 2020. And so this is why it's really important for us to continue to do more research on Beck-Fahrner syndrome to better understand all the clinical features as well as molecular changes. Individuals with Beck-Fahrner syndrome can share some craniofacial features as well. These can include kind of a tall or broad forehead, which you can see here. They can have flattening of the back of the head, which is called brachycephaly. They can have a longer than expected face, and a myopathic or hypotonic face where you see the downturned corners of the mouth, or an open mouth appearance. And that just illustrates it, that just shows the low muscle tone in the face. They can have protruding ears and a high arched palate. And then to help remember the features of Beck-Fahrner syndrome, we created this mnemonic using an abbreviation here that is from one of our genetic databases that names syndromes. So we can remember some of the features by this mnemonic. So behavioral differences, epilepsy, characteristic facial features, autistic features, hypotonia, retardation of development, and then size differences. Again, this doesn't include all the features, but it's a way to remember some of the key features. So what evaluations are needed when someone is diagnosed with Beck-Fahrner syndrome? So in essentially all cases, a developmental assessment is needed. And Dr. Harris and Dr. Eng will talk more about this, but this is to refer to appropriate developmental services. An echocardiogram is needed to do and this is an ultrasound of the heart to look for any differences in the way the heart was formed. Abdominal ultrasound, this is just to look for structural differences in the way the abdominal organs are formed, like the kidneys, the liver, the spleen, and others. An eye exam, because we know that individuals with this condition can have vision abnormalities as well as alignment issues for the eyes. And then an audiology evaluation, that's just a hearing test, which is often accompanied by evaluation by an otolaryngologist or an ENT specialist. And then on a case-by-case basis, I would say individuals that have concerns for seizures would need to have an EEG or an electroencephalogram, but not everyone would need that. A brain MRI, if there are certain types of neurological concerns. Orthopedics evaluation, if there's any concern for scoliosis or kyphosis or other musculoskeletal findings, and then others that would just occur on a case-by-case basis. And then lastly, DNA methylation testing is one thing that we do recommend in some individuals with Beck-Fahrner syndrome, which I'll talk about in a little bit. So how was Beck-Fahrner syndrome discovered, and how is it diagnosed? So it was discovered and is currently now diagnosed by using exome sequencing. And exome sequencing is a test in which all the DNA that makes all the different proteins is sequenced or tested to look at all the genes in order to identify the cause for an individual who is suspected of having a genetic condition. And in fact, many individuals with Beck-Fahrner syndrome, this is how they were diagnosed. Probably many of you are familiar with this. But this is showing here the first paper where we first reported Beck-Fahrner syndrome, though we initially called it TET3 deficiency. And we performed exome sequencing, so sequencing of all the genes, of one affected individual and her parents. And then we collaborated with clinicians and researchers across the world who had also done exome sequencing. And we were able to identify 11 affected individuals, shown here by the shaded circles and squares, from 8 families, and all had very similar clinical features, which I just described. And we found that all of these individuals had variants in the TET3 gene. And this is just a schematic of the TET3 gene showing the position of the variants that we identified in this first paper. And so in this paper, we wanted to really try to start to understand what might be going on in Beck-Fahrner syndrome. And so we tested many of the variants in TET3 in an assay in cells that we can grow in culture in the lab. And we showed that all but one reduced the ability of TET3 to perform the first step in erasing DNA methylation, so to remove this red 5-MC. And we found that all the variants that we tested, except this one here, we found that all these other variants reduce the ability of TET3 to be able to remove DNA methylation. And it was similar to a positive control, which we knew couldn't remove DNA methylation. And so this suggested to us that it is actually a deficiency in the enzyme that causes disease. So we then went on to identify a specific pattern of DNA methylation in Beck-Fahrner syndrome. And this DNA methylation pattern or signature can be used to diagnose or confirm Beck-Fahrner syndrome. And this is a complicated figure, but I thought it might be interesting for people to kind of see the types of data that we used in coming up with this DNA methylation signature. And so we hypothesized or thought that if TET3 was an eraser of DNA methylation and it was not functioning properly, that we might see an increase in DNA methylation in the blood of individuals with Beck-Fahrner syndrome. And so we performed genome-wide DNA methylation testing to see if we could see a difference in the DNA methylation present in blood samples from affected individuals with Beck-Fahrner syndrome when we compared those to samples from unaffected individuals or control samples. So this analysis shown here is a complicated, has a complicated name called hierarchical clustering, but basically it groups samples based on their similarity to one another. So each column here represents an individual patient sample. And there are 3 columns here under the red block and 3 columns here under the blue block, and then many, many columns here under the green block. So the red indicates individuals with Beck-Fahrner syndrome that have 1 variant in TET3. The blue indicates individuals with Beck-Fahrner syndrome whose samples have 2 variants in TET3, so 1 in each copy. And then the green indicates samples from control individuals who are known not to have variants in TET3. And then each of these rows, and there are 285 of these rows here, represents a site of DNA methylation in our genome, so in the whole collection of genes. And the color scale shown here, from dark blue to dark red, indicates the methylation level. So 0 is dark blue, and fully methylated is dark red. And so the darker the red, that indicates more DNA methylation. So what you can see is that samples from individuals with Beck-Fahrner syndrome, shown here under the red and blue blocks, have darker red and so have increased methylation compared to individuals without Beck-Fahrner syndrome or control samples. And another way of looking at this, which is maybe a little easier to see, is again another way of sort of separating the samples based on similarity. And you can see here that samples from individuals with Beck-Fahrner syndrome, shown in blue and red, are separated from control samples, shown in green, and that even samples that have variants in both copies of TET3 are separated from those samples that have variants in only 1 copy of TET3. So the 2 variants are shown in blue, 1 variant is shown in red. And so this showed that Beck-Fahrner syndrome does have a characteristic DNA methylation profile or pattern that can separate samples from individuals with this condition from those without the condition, or control samples. And this DNA methylation test is now a clinically available test called EpiSign, and it can be done in both the United States and the Netherlands. And we are also doing this test on a research basis to try to improve it. And this test can confirm a diagnosis of Beck-Fahrner syndrome. So we have multiple ongoing research projects, which Dr. Ji-Young Kim will discuss in more detail. But I just wanted to kind of briefly mention them, since I was just talking about some of the features of Beck-Fahrner syndrome and the molecular changes. So we are collecting additional data on the clinical features of Beck-Fahrner syndrome to better understand the disease and identify any other manifestations that might be there that we've missed to this point. Specifically, Dr. Eng, who will be talking a little later, will be performing detailed neuropsychological testing, or she can also collect data from previous testing that's already been done to better understand the neurological and behavioral features. And then we are also doing DNA methylation testing, like I just showed, on individuals with Beck-Fahrner syndrome and controls to improve our understanding of the disease and to improve diagnosis. And we are also doing some disease modeling and testing of treatments for Beck-Fahrner syndrome on brain cells or neurons and brain organoids that we can grow in culture. And again, Dr. Ji-Young Kim, who's working in my lab on many of these above projects, will be discussing these in more detail. And Dr. Rowena Eng will discuss the neuropsychological study in more detail as well. So in summary, Beck-Fahrner syndrome is a newly described genetic disorder due to variants in the TET3 gene. The TET3 gene encodes the TET3 protein, which is an eraser of DNA methylation. Beck-Fahrner syndrome may result from an inability to erase DNA methylation at many genes in the genome. And individuals with this condition have a specific pattern of increased DNA methylation on blood testing. Individuals with Beck-Fahrner syndrome have mostly neurological features, though they have additional features that affect other systems as well. And we recommend evaluations for all of these features when a diagnosis of Beck-Fahrner syndrome is made. And then we are doing research to better understand many aspects of this condition so that we can better understand it, better diagnose it, and hopefully someday identify a treatment for it. And finally, I just want to thank all the patients and families who have contributed to our research already. I especially want to thank Jamie Middaugh and the Beck-Fahrner Syndrome Foundation. Jamie, as I said, has done an amazing job thus far, and I look forward to working with her for a long time. And then I also want to thank our collaborators. I especially want to thank Dr. Ji-Young Kim, who's working in my lab, who's really leading this project currently, as well as our other collaborators who helped describe the condition, Dr. David Beck, as well as the collaborators that helped identify the DNA methylation signature, Beckham Sadokovic and Michael Levy, and then all of these other people across the globe who really contributed to identifying this condition and to identifying the DNA methylation signature. And then I also want to thank our funding sources that are funding this work in the lab. And then last, I just want to mention our multidisciplinary epigenetics and chromatin clinic at Johns Hopkins. So I direct this clinic, and I'm the clinical geneticist that sees all of the patients. Dr. Jacqueline Harris, who will be speaking next, is the pediatric neurologist and neurodevelopmental specialist. She also sees all of the patients in our clinic. And Ms. Jacqueline Britton is our genetic counselor who also sees all of the patients that come to our clinic. And then our medical office coordinator, Felicia Easley. And so anyone who comes to our clinic sees all of these providers at one time. And then we also help to coordinate appointments with Dr. Rowena Eng, who's speaking a little later, who is an integral part of our team, but isn't physically in our clinic each time we meet. And if you're interested in making an appointment, we would love to see you in clinic. You can either call this number in the US, so 410-955-3071, or you can email me. My email address is here. It's jfahrne, the number 1 at jhmi.edu. And I look forward to meeting as many of you as possible. Thank you so much.